Today we are learning about the introduction of the microbiology, means very basic terminology. In this lecture we have to focus for the microbiology, right? We are starting from to the very pioneering basic terms of the microbiology. Let's talk about the introduction of the microbiology. That is that one. First we have to understand about the word of the microbiology. Let's focus on it. Let's these conditions. Microbiology means micro means a small, bio means a living, and logy means a study. Means it summarizes all these conditions. It's called small living study. This is called as the microbiology. Clear? Robert Koch and Louis Pasteur, who gives a basic funda of the microbiology. The two scientists are given us a very pioneering form of the microbiological word, right? One, Robert Koch, who is invented about the bacterial and their characteristic, their growing patterns, media, their conditions, and Louis Pasteur, this gives us a many fermented factors and a sterilization process into the microbiology. In microbiological world, no any situations is possible without the sterilizations, right? Sterilization in the sense of the hospital factors, that clinics and that instruments and process with the patients. These all conditions which is related or correlated with the sterilizations, right? So these two scientists are involved a very much important to a concept, means the contributions into the microbiological world, right? Another scientists who are also involved and contribute their experiments into the microbiological world, but these two major scientists who gives us many types of the experimental factors and their characteristic of these eventical factors of the microbiology, right? Let's focus on to the other introductory factor of the microbiology and their terminology. Where it's coming from? Microbiological world, which is coming from to the 5 types of the kingdom, right? Who gives us to the Whitaker's 5 kingdom, where the microbiological world which is coming around. This is very basic terms to understand about the microbiology. This is very easy, students. So there are 5 types of the kingdom who already proposed by the Whitakers, which is going to be accepted. Out of that 5 types of the kingdom, only we are talking about the bacteremia, who gives us the knowledge about the any pathogenicity type of the bacterial infections, their characteristic and infectious factors from to the microorganisms. Clear, everyone? So this Whitaker's 5 kingdom. Out of that bacteremia we have to study, right? So the next one, what is it mean by the introductory factor of the microbiological factor? Bacteriemia, it indicated that many type of the pathogenicity type of the bacterial things which is observed into the microbiological world. Right now, this pathogenicity, what is its mean, the pathogenicity? This understand, we have to understand and focus of the pathogenicity means infections which is transmitted from to the pathogens. So now we are focusing on the 10 types of the pathogens that are, first one, that is the bacteria, virus, rickettsia, chlamydia, mycoplasma, fungus, then after spirochetes, helminthids, and another types of the 10 types of the disorders which is coming from to these types of the microorganisms. Let's written down on these notes. You are understand and focus onto this screen board. How many types of the microorganisms we have to study into this microbiology. Let's talk about that. There are 10 types of microorganisms are observed right now. The first one, bacteria, virus. Third one, mycoplasma. Fourth one, that is the fungi. Fifth one, algae. The 6th one, protozoa. The 7th one, spirochetes. The 8th one, this is the rickettsia. Ninth one, chlamydia. And the 10th one, this is the helminthids. These are 10 type of the microorganisms we have to focus by this in microbiological world. Right. Now let's talk about the first one, a bacteria. Means a bacteria which is a 5 type of the morphological characters we will be discussed into the next lecture. Now this 10 type of the microorganisms we have to focus by their own morphological characters and their pathogenicity. We are only observed onto the pathogenicity. Due to that reasons we are only focusing onto the bacteria, right, in your subject. Bacteria, then after another one, virus. Virus which is two types, envelope and non-envelope RNA and the DNA based, right. In the both formation which will be never been to observed of the virus in a simultaneous factors, like virus is always observed into the either RNA form either into the DNA forms, right. We are already discussed into your unit which is accepted from to your virolytic approach into unit number 4, right. Today we are only to understand about in these lectures for general introduction of the microbiology. The another one, mycoplasma factors, which is another cell wall characteristic which is belongs from to the pathogenicity. Fungi means this is the types of the fungus which is belongs from to the fungus, like the candidial factors, Candida albicans, pathogenicity type of the microorganisms which is coming from to this fungus, means this is opportunistic infections which is observed onto the human body. Only we are focusing onto the opportunistic fungus and their characteristic onto the funginemial factors. Another one, algae. This is the eukaryotical factors, algaetic aspects. This is the algae. Protozoa factor means transmissions from to the protozoa, means protons, like the way of protozoa. Example, malaria, which is transferred by the protozoal transmission disease. Spirochetes means which is observed as spirochetes, that morphological characters of the microorganisms which is observed by the spiral form. This is the spirochetes. Another one, rickettsia. This is another one characteristic, the types of the microorganisms or this pathogenicity which is transferred by the anthropod groups of the bacteria, means which is transferred by the vectors, right. This is the example, Rocky Mountain spotted fever. This is the rickettsial transmission. This is another one. This is the chlamydia. This is another one group of the bacteria, means this is the rickettsial and the bacterial in between. We will be fitted down this type of the characteristic. This is name is the chlamydial factor. Another one, this is the helminthids, means worms transmitted disease. This is called as the helminths factor. Helminths means worms transmitted disease, means tapeworms and the roundworms. I hope you understand about the 10 type of the different microorganisms who causes for the pathogenicity into the Homo sapiens species of the human being, right. Further, we are talk about the other introductory factor, means first we are studied about the 5 type of the bacterial morphology, means morphology of bacteria. So there are 5 type of the morphological characteristics which is observed into the bacteria, right. This is just general information. What type of the subjects and what type of the subtopics we will be discussed into your remaining, means your continuity type of the lectures. This is the general concept and introductory factor of the microbiology. How? Means every subject have their own terminology, means nomenclatures, their understanding purpose, their medical terminology. So in this subject, microbiological world have also that a terminology, means specific medical terminology we have to understand. Specifically for we are talking about the bacteria. Bacteria which is always observed into the 2 types. This is the genus name and their species name. Means we are talking very from to the basic example. Let's written down how the bacteria will be interpreted on these conditions and their how to interpret it by the genus and the species characteristic, right. Let's talk about first example. I am taking one example with you, how to written down and how to understand about the condition of the terminology of the bacteria. Let's give us one example. Terminology. Terminology which is observed into the 2 types. Means a bacteria which is always observed into the two types. First one, this is the genus name, and second, that is the species name. Genus and the species name. Right? I hope you understand about that things. Right. Let's talk about that example. Means a Mycobacterium tuberculosis. This is one example of the bacterial name. Right now, this is a cause for the TB. Are you getting my points? I hope you. So, in these conditions, we have to elaborate about this species or the genus. How to be interpreted like that way. Mycobacterium, this is the genus name, and this is tuberculosis, this is the species name. Means this is the genus name, this is space, and this is the species name. Species always differs the disease. Means ki every species which is always differ for that causing for the disease conditions. Right. The genus, this is a Mycobacterium. This is the genus name. Means this tuberculosis bacteria which is coming from to that genus name, that name is the Mycobacterium. Right. And this is the species name. Species always differ the different disease. If genus is same but the species will be going to be changed, it indicated that the disease will be changed. That example, let's talk about that. Mycobacterium lepra. Right. This is the genus name and this is the species name. Lepra caused the leprosy. Lepra caused the leprosy. This is the genus name and this is the species name. Clear? I hope you understand well. Right now, I am giving just basic example of the terminological condition. Your father and mother is your parents. That parents always cause for the genus, and you and your brother sisters which are your species. So, you and your brother sisters never been to your habitat and that common things are observed. So, the genus is same, but you and your brother sisters which is going to be their naturatic aspects and their habits are changed. So, in this way, we have to focus on it. This basic terminology you can understand, that put it down into this terminology, that is genus name and the species name. Now, how to be written down of the genus and the species name of the bacteria? Let's talk about that. Mycobacterium tuberculosis. We have to underline of this whole bacterial name, means, or we have to written down like a first letter which is going to be always in a capital letter, dot, and the species which is always written down into a italic letter or into the second letters. Right, like that way. Clear? This is caused for the MTB. Right. Either you have to written down like that way, written down whole genus and the species name and causing for the underline, or another one, that is things, just because of these reasons, this M is capital, dot, M indicated the capital M letter which is a capital letter which is indicated a genus name, and the tuberculosis we don't need to underline of this genus or the species name. This is the genus name and this is the species name who causes for the TB. Right. Another one, let's give the another example. Mycobacterium lepra. We have to written down like that way. This is underline. We have to measure like that way. Or M is capital, dot, and lepra, who causes for the leprosy. Who causes for the leprosy. Either you can use this terminology, either you can use like that with this terminology. Clear? Let's talk about the first type of the bacteria and their characteristic. Let's focus on it. I hope you understand about the terminology and very basic introduction of the microbiology. Right, the bacteria have the 5 types of morphology. First one, bacilli. Second one, cocci. Third one, short rod. Fourth one, spirochetes. Fifth one, flagella. Clear? This is the 5 type of the morphological characters which is observed into the bacterial world. Right? Let bacteria always are measured in micrometer. This is the unit name. This is the micrometer. Micrometer which is always observed into the bacteria. Clear? Another one things. Bacteria always observed under the oil immersion lens. Always observed under the oil immersion lens. Right? Means a oil which used into the oil immersion lensing factors. Right? This is the unit name, this is the micrometer, and the bacterial which is observed under the oil immersion lens. Right now, we are focusing on to the bacterial and their morphological characters. How the morphological characters which is differ by their characteristic. First one, bacilli. How to be interpreted of the bacilli? This is the outer morphology. Morphology which is called as the anatomical factors means ki outer observation. This is called as the morphology of the bacteria. Anatomy of the bacteria, outer structure of the bacteria. I hope you are understand, students. We are indicating always morphological character in a round factor. Why? Because of which reason the lens of the microscope, this lens is always appeared in a which shape? This is the round shape. Due to of that reasons, we are always observed in a round manner. Right? I hope you understand, students. Bacilli means bacilli which is always observed at their morphological characters by the rod shape and always arrangement into the chain or in the pair. Like this is the rod shape. This is always observed in a pair. Like this is the pair. This is the arrangement. Means a shape. This is the rod shape. Arrangement. In a pair and in a chain. This is always arrangements and pair of the bacillus factor. Another one, this is the cocci. Right, cocci, so which is around. This is a microscopical factor. Cocci which is always observed in a cluster and a round. Means this is always observed into the rounding factor. Means a shape, round and arrangement, cluster, group. This is the arrangements. Right. The third characteristic, that is the short rod. The third characteristic which is the short rod. Again we are to focus into the round manner and the shape in a rod shape and arrangement into the singly. Arrangement which is always observed under the single, like that way. This is the rod shape. This is arrangement and the shape of the bacteria. Right, let's quick revision between these conditions. Bacilli, the shape is always, this is a 2D observations by the microscopical image. Whenever we are talk about the identification of the bacteria, the very first step, that is we have to focus on the morphological characters, and the morphological characters would be always observed by the staining factors, staining condition. After the using the stain, after applications on the staining patterns, we will be observed under the oil immersion lens, and that images will be going to be observed in a 2D dimensional factor, like that way. Whatever I am focusing on it, and I am written down already on it. Right. This is the 2D image of the bacillus factors. Right. See, this is the quick revisions we have to follow about that thing. So we are remember for the long term in memory. Right. Quick revision. See, this is the in a chain or in a pair, and the shape, that is the rod shape. What is that shape? That shape is always observed in a rod factors. Cocci always observed into the round and into the cluster. The shape which is a round and their arrangement, that is in a cluster. Short rod, the shape is in a rod shape and arrangement which is observed into the singly, means ki that is the rod shape and the singly. These are all 2D images. Now we are focusing on to the how it's actually observed into the 3D factor. Let's zoom it out. See, this is in a oval shape in a 3D form. This is the microscopic image after the stain. This is the rod shape, but in a 3D factor which is oval shape and into the chain. As usual, in this zoom factor 3D, the 3D factor which is observed into the cluster, like that way. This is the 3D observations. Another one, that 3D factor which is always observed in a single image key into the oval shape. These are the short ones. Okay, this is the 3-1 is completed. Now the fourth characteristic, fourth morphology. The fourth morphology is the flagella. They have the 4 type of the flagella. First one, this is the bacteria. It's always give to the movement to the bacteria. Okay, flagella. This is called as the flagella. This is a one type of the flagella. This is the second type, means ki one pole, both the pole, multiple at one ended side, and overall on the body. There are 4 type of the flagella factors which is observed into the bacterial world. The next one, which is the 5th one, Fimbria or Pilia. What did it mean? Means ki this is another characteristic we are also observing like that way. Not for the 5th, let's talk about the 4B, 4A. This is the Fimbria and the Pilia. What is that? This is the bacterial cell wall, and this is small appendages factors which is observed onto the bacterial cell wall. This is all called as the Fimbria and the Pilia. These all are used for the multiplication, means ki this give for the bacterial reproduction. Bacterial reproduction. How it's possible? Let's talk about them. This is bacteria. This is called as the Fimbriator factor. The small appendages observed outer the bacterial cell. Then after, this is another bacteria, this is small also, appendages observed on it. Right, so that way of the things. One, this is appendages which is attached to the another bacterial appendages. This is the prokaryotic cell. This is example of the bacteria. This is a genetic material which is absorbed into the DNA in a circular manner. Only this is the prokaryotic factor, not eukaryotic, by mistake. This is the prokaryotic cell. This is two bacterial example is there. These two appendages which is attached by each other and transform the genetic material from one bacteria to the another bacteria. This is called as the F plus and this is called as the F minus. Main purpose is that what is the utilization of the fimbriatal manner which is observed onto the bacterial cell wall, right? This small appendages which is attached by the another one type of the bacteria, the genetic materials, one bacteria which is transformed to the another bacteria, this is called as the F plus, and who is gaining, this is called as the F minus. Who is donating, this is called as the F plus. Who is receiving, this is called as the F minus. F is equal to fertilization. This is called as the fertilization, right? These three types of the transforming of the genetic material which is observed means key connections which is a conjugation, transduction, and third one transformation. These three types of the mechanism which is observed in this bacterial reproduction, right? Conjugation means ki what the type of one appendage is which is attached to the another one and this a bonding factor made, this is called as the conjugation. The genetic materials who is going to be transferred by this conjugated bond to the donor factor to the receiver, this is called as the transformation, and the budding process will be successfully done like that way in a bacterial manner. This is called as the transduction. Clear? So, this budding factor, what is called as the budding? Means in a prokaryotic, especially for a bacterial divisions which is always observed by the budding, means this way the prokaryotic cell wall division, this is always in a unicellular division. In a vice versa, eukaryotic cell divisions which is observed in a multicellular division. This is a prokaryotic and this is the eukaryotic, means this is a multicellular factor. This is eukaryotic cell division. Clear? The another one, that is the 5 type of the characteristic, and the last one, 5th one, spirochetes. Spirochetes means ki a bacterial cell wall which is observed into the spiral form. This is a spiral. This is the coils and the spirals which are observed onto the spirolytic factor. There are 5 type of the morphological characters which is observed into the bacteria, right? Another factor, bacterial. Bacteria have two type of cell wall characteristic. First one, a gram positive, another one gram negative. This is the difference between a bacterial cell wall. Whenever we are talking about the bacterial cell wall, we have to interpret it about the gram. Gram which is indicated there, the name of the scientist, we have to give a name whenever we are talking about the bacterial cell wall. Like this is the gram positive, another one the gram negative, right? The difference between a gram positive and gram negative, let's talk about that. A gram positive cell wall characteristic have the thick, and this cell wall is the thin. Due to which reason? This is the characteristic gram positive and gram negative. Let's talk about them. First one, this is the thick and first one thin. Due to which reason? A periplasmic space. What is called as the periplasmic space? This is the cell membrane and these are the arrangement behind cytoplasmic factor under to the cell membrane. This is called as the periplasmic space. Due to the characteristic, it is always observed into the thick manner. And what is called as the thin means ki the arrangement like in this way. The periplasmic space is completely not observed, means ki we are not avoiding, means ki 100% we can't say like that, but we are get same, means ki the characteristic of the periplasmic space is very less or not possible. This is due to the reason that arrangement of this bacterial cell wall, it's observed into the thin. Clear? The next one, the bacteria after the staining, the bacteria observed of the gram positive, that is the bacteria. I am talking about the bacteria after the staining. Let's talk about that. After the stain, bacteria would be observed in a purple bluish color and background is reddish pinkish color. Reddish pinkish in color. Clear? I hope you understand well. Second, gram negative. After the stain, bacteria observed, means ki bacterial color after the stain, bacteria observed into the reddish pinkish and the background is bluish purple. Right, this is the major difference between the gram positive and gram negative bacteria. The next one, what is its mean? Let's talk about that. The third characteristic, teichoic acid. This is a specific type of the protein, teichoic acid, which is present in only and only into the gram positive bacteria. And the third one, mucopolysaccharides or lipates are observed into the gram negative bacterial cell wall. Clear? This is the basic difference between the bacterial cell wall, that is the gram positive and gram negative characteristic. I hope you are understand about the introduction of the microbiology and terminology of the microbiology and also morphology of the microbiology. Okay, students?